The name of this experiment is Demonstration of how to prepare an agar slant. Slanting the surface of the agar gives the bacteria a greater surface area on which to grow in a test tube. Agar slants are useful in maintaining bacterial cultures as it requires less space. The requirements to perform this experiment are a source of bacteria, inoculation loop, nutrient agar media, test tubes, autoclave, and laminar airflow. To start this experiment, the first thing we need to do is preparation of the nutrient agar media. For the preparation of nutrient agar media, we will be requiring distilled water. So we take 500 distilled water in a conical flask. After that, we need to weigh the media as per the requirement of the 500 ml of distilled water. So here, using a weighing machine, we are weighing the nutrient agar media. As per the requirement of we need a media of almost 6.5 grams. We take a little bit of extra so that it compensates the media which would be sticking to the paper on which we were using for. We need to scrap the remaining media left on the paper. As the complete media, powder, nutrient agar powder is added to the distilled water, turning it into a homogeneous solution. So in order to avoid the nutrient media from remaining in state, to add a little bit of extra agar to that solution before we take it for sterilization. So here we are adding some extra amount of agar to the nutrient media. We'll mix this again, mix it well again. As we finish mixing, cotton plug, it must be tightly covered. Next, we need to sterilize the media that we have prepared. The sterilization process is done using an autoclave. So here, after checking the amount of water, we place the flask inside the autoclave, close the lid properly, and then we switch on the power for it to start. We'll wait for around 15 to 20 minutes for the pressure to come up, and so that the sterilization is over. So here we see after 15 minutes that the autoclaving is over. So we then switch off the power and let the power release gradually, and by that time we would clean the working place. We would be requiring the laminar airflow. So before we start our work, we sterilize the area. We sterilize the working bench of the laminar airflow as well as the wall. The wall is done using ethanol. We rub properly using cotton. Then we switch on the blower for some time. In order to completely remove all the contaminants from inside the laminar airflow, we need to switch on the UV light. So prior to that, we wire things into the laminar airflow so that everything gets sterilized well. So here we have placed our petri plates and from the hot air oven. After sterilization, we have also kept the spirit lamp as well as the inoculating loop inside the laminar airflow for their sterilization also. We now switch on the UV light and let it work for 15 minutes. As the 15 minutes, we also check the autoclave. We find the pressure has lowered down, and now we would open the autoclave and take our out of that. We need to handle it carefully as it is very hot. So this is our media which has been sterilized, and we will be carrying it towards the laminar airflow. By now it's 15 minutes is already over, so we switch off the and then we put the media inside the laminar airflow. Media is poured into the slant. When the agar is still hot, carefully tilt the rack holding the test tubes on a solid surface, making sure the medium inside the tube is at a slanted position relative to the test tubes. The medium is allowed to cool and solidify at this angle, which increases the surface area of the agar. The slants are ready for use once the agar has solidified. Once the slant is solidified, we will now inoculate the slant by transferring cells to the inoculating loop. The inoculating loop is sterilized in the spirit lamp before the inoculation is done. The loop is moved across the surface of the slant and the tubes then recapped. The slants are out. On observation of the slant after 24 hours, there were evidence of growth seen inside it. The growth showed pure colonies, and therefore this agar slant may be useful in maintaining this culture for future use.